Question number 156. If a 4 kilo base pair fragment of DNA is to be amplified from 8 kilo base pair plasmid, then how many copies are obtained at the end of 30 PCR cycles approximately? And the options are 1 billion, 1 million, 2 billion and 2 million. And we know that in PCR it is possible to generate 2n molecules after n is what? N number of cycles. So after 30 cycles, this would be 2 to the power 30, and which is approximately 1 billion. So the correct answer to this question is option number 1, 1 billion. Rest of the options are incorrect, and this data is just to confuse you. So the correct answer to this question is option number 1. Let's discuss our next question. Question number 157. Part of the plasmid PBR322 that controls copy number of foreign DNA linked to it is, and the options are rope, multiple cloning sites, selectable marker, origin of replication. And the correct answer to this question is option number 4, origin of replication. It is a part of plasmid PBR322 that controls copy number of foreign DNA. If you want a high copy number of your DNA, the nucleotide sequence of ORI, this is ORI or origin of replication, is responsible for controlling the copy number of plasmid as well as linked DNA to it. Let's discuss rest of the options. The very first option given is rope, and this is an incorrect option. This rope, as you can see here, codes for the proteins involved in replication of the plasmid. The next option is multiple cloning sites, and this is an incorrect option. Multiple cloning sites or MCS is a polylinker, a short segment of DNA which contains multiple but unique restriction sites within the plasmid. So this is an incorrect option. And the next option is selectable marker, and this is an incorrect option because selectable markers identify and eliminate non-transformants from those which are transformants. So the correct answer to this question is option number 4. Let's proceed to the next question. Question number 158. A single PCR reaction involves 3 temperature-dependent steps. Their correct sequence is, and the options are annealing, denaturation, amplification. Second option is denaturation, annealing and extension. Third option is extension, annealing and denaturation. And the last option is annealing, extension and denaturation. And from this given diagrammatic representation of a PCR cycle, you can see that this double-stranded DNA is denatured first at the first step known as the denaturing stage, followed by annealing stage at 50 to 56 degrees centigrade, followed by extending stage. So the correct sequence would be denaturation, annealing and extension. So the correct answer to this question is option number 2, denaturation, annealing and extension. Let's discuss rest of the options. The very first option given is annealing, denaturation and amplification, and this is an incorrect option because denaturation is followed by annealing and extension or amplification. The next option is extension, annealing and denaturation, and this is an incorrect option because denaturation is followed by annealing and then by polymerization or extension stage, and the last option is annealing, extension and denaturation, and this is also an incorrect option. So the correct answer is option number 2. Let's proceed to the next question. Question number 159. Treatment with salts of which ion facilitates efficiency of transfer of foreign DNA into host cells? And the options are oxygen ions, calcium ions, potassium ion and sodium ions. And the correct answer to this question is option number 2, calcium ions. Calcium ions or divalent cations causes the DNA to precipitate onto the surface of host cells, and thereby enhancing chances of entry of recombinant DNA into host cell at 42 degrees centigrade or heat shock step. So the correct answer is option number 2. Rest of the options, oxygen ions, potassium ions and sodium ions, are the incorrect options because we need divalent cations for it. So the correct answer to this question is option number 2. Let's proceed to the next question. Question number 160. Recombinant DNA is directly injected into the nucleus of an animal cell. This method is known as, and the options are transformation, microinjection, ballistic, and electroporation. And the correct answer to this question is option number 2. Microinjection is a method by which recombinant DNA is directly injected into the nucleus of an animal cell. Let's discuss rest of the options. The very first option given is transformation, and this is an incorrect option because transformation is a procedure through which a piece of DNA is introduced into a host bacterium. The next option is ballistic, and this is an incorrect option because in ballistic, or also known as gene gun method, cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA. The last option is electroporation, and this is an incorrect option. In electroporation, short electrical impulses of high field strength are given to the target host cells. So the correct answer to this question is option number 2. Let's proceed to the next question.